Hi, I'm Olivia Alcantara. I'm an undergraduate student of Pharmacy and Scientific Initiation student at the Laboratory of Bioinformatics and Molecular Modeling, LABI, at the Federal University of Bahia. Today, I will present to you my work entitled Bioactive Compound Selection for the SARS-CoV-2 as Glycoprotein, carried out under the guidance of the Dr. Samuel Pita. The COVID-19 caused by SARS-CoV-2 remains a serious health problem worldwide. It's responsible for the current pandemic state declared by the World Health Organization since March 2020. Until now, the cumulative numbers of deaths are more than 4.7 million and over 231 million of infected in the world. We already have some drugs approved for its treatment, but they have a lot of limitations. Considering this, the search for antivirals to treat COVID-19 is tense. In this subject, the virus spike S glycoprotein has been considered a promising target for drug development. The S mediates the virus entry into human cells through the interaction with angiotensin-converting enzyme 2 receptor. Thus, it is work with searching bioactive compounds against S through virtual screening. One protocol database was used in our study. In my sketch, we draw the structures and calculated its physical-chemical properties. Next, the enzymatic properties were analyzed in several servers, such as PGR, Factors, Aggregated Devices, SwissADME, DataWarrior, and PKCSM. The 3D structures of the ligands were optimized in the software HappyChem. Finally, we used Autolog to adjust parameters, such as addition of hydrogens, calculation of density charts, and set the atom types. Spike structures are selected from protein data bank, analyzing FTMF for durability. The structures of 6VIV and 6ZGG were prepared in Autodoc by removing water, adding polar hydrogens, calculating density charts, and adjusting the atom types. The docking was performed on 3 different sites of spike according to durability results, where site 2 and 3 were located in the RBD region. All parameters remain the same under the docking calculations. Protocols were chosen due to the wide range of bioactivity properties that they may present. In addition, there are evidence in the literature that some compounds show activity against S and Pro of SARS-CoV-2. Regarding their physicochemical properties, they meet at least 3 of Revitsky's work criteria, which indicates that they are drug-like and orally bioavailable. The toxicity prediction made by Delta Warrior indicated that the ligands had low potential to cause toxic effects, such as teratogenicity, mutagenicity, irritation, or being tumorigenic. Also, most of them were non-cardiotoxic and had low potential to inhibit liver enzymes that are involved in drug metabolization, which is important to predict drug interactions. So, through the drugability analysis, we identified an unconventional druggable site. On spike, a central region had 3 clusters with a total of 40 probes, which can be considered a druggable site. These sites were then applied on our docking studies as site 1. Through docking, we analyzed the interaction between S and catechols. We used a total of 383 molecules. Here we have presented the best ligand results. There were the MFCT 17654, 17746, 7418. Those molecules also presented good ligand affinity with ligand efficiency bigger than 0.29 and lipophilicity corrected ligand efficiency between 10 and minus 10, as described by Hopkins. About the interaction data, we observed that on site 1, the ligand interacted with glutamine 1002 and 1010, threonine 1009 and 6, and alanine 766 of the chains ABC with hydrogen bond type interactions and hydrophobic interactions. These results corroborated with the data from Ugar and Mayo that also used the site. In site 2, in RBD, the ligands interacted with amino acids that are involved in spike human receptor interaction, such as glutamine 493, tyrosine 495, and lysine 417. The same did not happen on site 3. So, as conclusion, we have that all data were useful because they were able to identify promising chemical molecules with favorable interaction energies and affinity ratios, such as ligand efficiency and lipophilicity correct ligand efficiency. We are also able to identify new druggable sites in spike, as well as important results for the interaction with the ligands. Thus, we hope that our results would aid to drug development for the COVID-19 treatments. We also would like to thank Brazilian National Council for Scientific and Technological Development, PIBIC, UFBA, and LABIN for my scholarship. Those are the references that we used. Thank you very much for listening.